Good afternoon, CGIL. This week we are pleased to welcome Dr. Henrique Mulim presenting for us. Henrique is an animal scientist specializing in genetics and animal breeding with a strong background in livestock research. He earned his master's from the State University of Ponta Grossa, where he studied genotype-environment interactions in Holstein cattle, analyzing how environmental conditions influence genetic expression. He later completed his PhD at Federal University of Bahia under the supervision of Victor Breno Pedrosa, focusing on genetic diversity, the genomic characterization of the Puruna breed, and heterozygous rich regions. In 2024, he joined Purdue University as a postdoctoral research assistant under the supervision of Dr. Inaya Rojas, researching the genetic diversity of the Angus population worldwide. His work contributes to the understanding of breed development, conservation, and the future of livestock genetics through research and innovation. Today, he will be discussing the genetic puzzle of Angus cattle, selection, inbreeding, and diversity. So go ahead whenever you are ready, Henrique.
Okay. First of all, thank you so much for the introduction and also for the invitation. So just to, I would like a heads up, can you hear me well and also see my screen well?
Yes, we can hear you and the screen looks good.
Okay, thank you so much. So today I'm going to present a little bit about the project that I have been developing this last year here at Dr. Rojas' lab, where we are working with some Angus populations for different parts of the world. So I entitled the presentation as the genetic puzzle of the Angus cattle, selection, breeding, and diversity. For the presentation, I split the presentation in 4 parts, where we're going to talk first about the history of the Angus cattle, so you can have an idea how this breed was selected over time, and also the importations in different countries. The genomic structure of the Angus cattle worldwide, so we can see and compare how the diversity of these animals and how they are similar or different. And more specifically, in the third part, we're going to talk about the inbreeding depression in Brazilian Angus cattle that we measure inbreeding based on the pedigree and also based on the genotypes. And our last topic, I'm going to present a little bit about the projects that we have been work and our members from the Dr. Rojas labs. So let's talk a lot about the history. So Angus cattle that has the history linked to the Scotland is a Scotland breed that was bred over 400 years. And one interesting points about the breed is that they have adaptability to various climates and environments, and also naturally polled and a high quality beef. This is one of the main advertisements that when you talk about Angus is the high quality of the meat, and that came with the history of the breed. So no, we don't have only the black Angus, although is the first animal was the black. We also have the red animals. These also track that have their orange track back to Scotland, and it suggests that the red color was originated in the 18th century when they crossed the black pulled cattle or the Angus cattle with the English longhorn cattle. This was with the intent to improve the size and the strength of the animals, while the offsprings were typically black. They carry the recessive red gene. Just for curiosity, this red color is linked to the MCR1 gene, or the melanocortin-1 receptor gene, that is going to be found in the chromosome 18, and is just a mutation, missense mutation, a change for a substitution of a leucine by a proline, resulting in the red color of the animals. When you're talking about the animals in the history, we're going to see that during the 20th century is where we're going to have more the intensity selection happening in the animals. based on the growth rate, the body size, and feed efficiency. So the animals start to be more popular and spread around the world. Talk more how the animals was spread around the world. Can see that the first documented importation happened in Australia in 1940, where 80 animals were imported and brought to Tasmania. On there, the breed quickly became popular among Australian cat producers, basically because of the high quality of the beef, but also because the adaptability to the Australia's diverse climates. With the popularity among the breeds in 1946, the Australian Angus Association was established to promote the breed and also help the farmers. For United States, we have the father of the Angus breed here in the United States was George Grant. When in 1973, he imported 4 Angus bulls from Scotland to Kansas, and he crossed these animals with the Texas Longhorn cows that they had here at the time. What they saw was that the progeny, they have a high quality beef, strong maternal abilities, and also climate adaptability, especially for the winter here in Kansas. In 1883, the American Angus Association was established once the breed started to gain popularity here, and to promote the breed and also to help the farmers. Until 1917, the American Breeding Angus Association, they registered together black and red Angus cattle. But in 1917, the association started to restrict the registration just only to black Angus to preserve the pure black lines. With that, the number of the reduce of the red Angus in the herds was reducing. So 7 breeds founding the Red Angus Association to promote the line and to have a specific breed line of the Red Angus breed. For Canada, we have a similar introduction and growth as United States. The first document importation about the Angus is found to be in 1860, when few animals were brought from Scotland to Quebec. And in 1905, the Canadian Angus Association was established, same way to promote the breed and also help farmers. Brazil, I think here about the history is the youngest in association. The first Angus cattle and also have different history and start to come from Scotland. The Angus that we have in Brazil, they came from Uruguay in 1906 and was important to Rio Grande do Sul, that is the southwest states on Brazil. In 1963, the Brazilian Angus Association was officially established, and to promote and help the farmers too. One interesting point when you talk about Brazil is that we have huge parts of their material important from United States, Canada, and also there is a relationship between United States, Canada, and Australia. So taking that information in account, we start to have some questions when you're talking about the history of the Angus cattle. First, with these connections between the countries or the importation of between the genetic material, how connected are these populations? And also, what is the impact? Because although we have this genetic exchange between countries, the selection that is made is totally independent between the countries. But what is impacting that this selection on the genome of this population? So we perform the first studies that we entitle Genomic Structure and Selection History across Angus population worldwide. We're going to talk about insights from runs of homozygosity, selection mapping and function analysis, but we also perform an analysis more related to the specific genetic structure of each one of the populations. So Angus, we know that Angus play a vital role in the beef industry, especially in countries that we have as the United States, Canada, Brazil, and Australia. And in these countries, they still applying the genetic and genomic selection methods to improve their performance in both feedlot and pasture systems. Thinking about the history of each one of the countries and their associations, our goal with this study was to verify the population stratification into one of this population: American, Australia, Canadian, the red Angus of America, and Brazil. Identify and classify and also see the patterns of runs of homozygosity inside of each one of this population. Characterize the genomic similarities and the difference among populations, and also verify the selection history using the generation proxy selection mapping to see the selection swap that we have inside of each one of these populations. For the maturity methods, we use 71,000 Angus genomic information, 15,000 Black American Angus provided by the American Angus Association, 15,000 Black and Red Canadian Angus provided by the Canadian Angus Association, 15,000 Black Australian Angus provided for the Angus Society of Australia, 12,000 Red America Angus provided by the Red Angus Association of America, and more than 13,000 Black and Red Brazilian Angus provided by Brazilian Angus Associations. The analysis that we performed was the principal component analysis in phylogenetic tree, so we could access the structure of the populations. Consists of gametic phase, runs of homozygosity, and the generation proxy selection map. For the principal component analysis, phylogenetic tree consists of gametic phase. We use only the markers in common between all the populations. And for the runs of homozygosity and generation proxy selection method, all the analysis was performed inside of each one of the population using the markers available for each one of the population. So the first results that we see in about the population stratification is we have two clusters of the animals. They group in two clusters. First, the three first PCAs they explain around 14% of the variation inside of among the individuals. Canadian, American, American Black Angus America, Brazilian and Australia, they group together in a unique group, and the Red Angus of American Red Angus, they all group together in a separate clusters. This tells us that although this population, they all share the same tracing back to the Scottish Angus, the selection performed over the time may have contributed to the difference that we observe here for the Red Angus association. On the other hand, the exchange material that we have between the countries, Brazil, Australia, America, Canada, and they can make, they can be contributed to the similarities among these individuals, made them grouping together. Same thing was observed for the phylogenetic tree, where the animals, they were more America, Canadian, Brazilian, and Australia, they were more together in the phylogenetic tree. When you end the Red Angus of America, they were more distance when you're talking about the distance in these populations. Although not presenting here, we also look for their metrics, for example, for FST or index fixation index, and they also showing that all these groups, the first group, that is the American Angus, Canadian, Brazilian, and Australia, they all have similarities, really strong similarities when they compare with the red Angus, where we're going to see more difference between each one of the populations. For the consistent gametic phase that we are analyzing the persistence between the LD and the KTLs inside of each one, the population. What you observe, although here I'm just presenting about the American with the other populations, we saw that for the cluster, the group America, Canadian, Australia, and Brazil, they always gonna have highest correlations inside of the persistent gametic phase when compared with the red Angus of America. This also is important if you're gonna talk about the genetic evaluation worldwide that we're thinking. Showing that these three populations, they have higher consist of a gametic phase. That doesn't mean that we cannot use, for example, the red Angus of America, but what it means is that we will need to have a higher number of markers more close to the KTL so that we can have this linkage, linkage disequilibrium between the markers and the KTLs more in common between all the populations. For runs of homozygosity, what we see, although we are working with 5 populations, I'm going to present just the 4th, America, Australia, Brazilian, and red Angus. For Canada, we have a problem with the genotypes, and during the quality control, we are removing a lot of markers. With that, we decided not to show the results for the runs of homozygosity once they were being affected by that. Just the runs of homozygosity. The others we are going to present. But as we saw that the results was not what we think it is happening in the population, the co-authors and I, we decide to not showing the results for Canadian Angus, Canadian Angus, sorry. But what we see is that for all the populations, we were able to observe smaller lengths of runs of homozygosity. And if you wanted to classify this length, we can say that this length, they are created around 25 to 50 generations ago. So with Brazilian and red Angus, we have a higher percentage of the runs of homozygosity found comparing with the America and Australia. And if you see here, all the populations, they also find higher, longer, runs of homozygosity. These runs of homozygosity around 16 megabases, we can classify that these can happen or were creating around 3 generations ago. So we still can we are able to classify when these runs of homozygosity are happening based on the time that they were created, based on the length that they have. But in the majority of the case, in all the populations, we have that many of our rates, they are formed in more ancient generations. More a particular opinion, more than just classifying and observe runs of homozygosity, I like to see the concentration inside of the population, so we can see signs of selection signatures. For the 4 populations, we observed that just only America and Australia, they have regions that pass the threshold of 50% of the markers falling inside of the ROH. So for the others, they are in some regions that are really close to the thresholds, for example, Brazil. In these islands, we identified 2 islands for American Angus in the chromosome 8 and chromosome 13. And in Australia, we just identify in the chromosome 8, being like in the chromosome 13, really close to the threshold. So almost like 2 lines of two islands being identified here. Looking for where these islands are, what is these islands are related, we find 7 genes inside of these regions. And all this relates to the tender score, body weight, subcutaneous fat, thickness, average daily gain, dry matter intake, and weight gain. So these are traits that have been considered in some point inside of the selections when you talk about the breed. These we found in the literature. And we can see that the presence of this mark of selection in the genome of the animals. Related to the generation proxy selection mapping, the only population here that didn't have any significant marks that you could see the change in frequency over the time was the Brazilian, probably because of the youngest group that we have here of the animals. So we didn't have the opportunity to see the whole change frequency over the time. But all the other populations, we identify markers that were significant for this selection proxy. When looking for what these markers are related, although each one of the population they have some particularities, we still found in general that these populations, these markers, they were related to body weight gain, maternal traits, growth, and equality. So these finds, they demonstrated that selection efforts by the associations, they have successfully altered the gene frequency or the allele frequency linked to these markers, and these markers are linked to economical and important traits. With this, the first conclusion that we have for this first paper was that we could observe genetic similarities among the American Angus, Australia, Canada, and Brazil. A distinct profile for the Red Angus Association, or the Red Angus animal, where they can sometimes be even considered as a different breed. The identification of runs of homozygosity to economically significant regions. So we observed that two islands, especially for America and Australia, we have signs of these selection signatures, and also evidence of the current selection strategies used by the Breeders Association. Talking about that, we are going to the second study where we are more close to work with the Brazilian Angus Association, where we were exploring breeding depression in Brazilian Angus cattle populations, pedigree, and genomic data set. When you're talking about the inbreeding, since the Darwin's earliest work in plants, comparing self-fertilized plants and plants that progenies that was coming from parents unrelated, the impact of the inbreeding has been studied in various species. Normally, when you talk about inbreeding, people sometimes associate just the inbreeding, the defects related to the inbreeding that is going to affect the fitness of the animal. But also inbreeding can affect all types of traits if they have some dominance effects on that. So inbreeding depression is going to refer to the decrease in the mean performance due to the increase of levels in inbreeding. To talk about what it is, to talk about the inbreeding depression, we need to define what is inbreeding. We have the first definition of an inbreeding was the correlation between homologous alleles of two gametes that unite to form individual by writing in 1922. Later, Maleko in 1948, he proposed that inbreeding actually is a probability that two homologous alleles at a given locus are identical by descent. Traditional, the inbreeding is measured based on the coefficient of the inbreeding was estimated based on the pedigree. But with the development of these NIP panels, new metrics start to be to create to estimate what we call inbreeding based on the genomic information. The problem is some of these, all these metrics, they normally are linking to the definitions of right and molecule. So we have two different definitions. And these methods, because they are related to these definitions, they bring different results. And right now, we still have the debate ongoing in which of these metrics is the best to capture inbreeding. And we don't have yet this answer. So seeing all the inbreedings that we have developing until now, our goal with this study was access 6 different metrics of inbreeding coefficient, estimate the inbreeding coefficient for each one of these metrics, using each one of these metrics for 13 traits currently recorded by the Brazilian Angus Association, and later perform what we call genomic-wide inbreeding depression analysis, where we're going to use the runs of homozygosity information to map possible genomic regions associated with the inbreeding depressions in these different traits. For the material and methods, we use the genomic information of 13,777 Brazilian was born between 88 to 2023, and genomic information of 17, more than 78,000 markers. The phenotypic data, we use 13 traits that are officially evaluated by the association, the Brazilian Association, and for the pedigree, more than 500,000 animals was accounting this pedigree. The 6 different metrics of inbreeding were estimated based on the pedigree information, based on the genetic relationship matrix. That is the first metrics method from Van Reyden. Based on the homozygous genotypes observed and expect, based on the homozygous genotypes, and based on the correlation between uniting gametes. And the last one was based on the runs of homozygosity. And inside of the runs of homozygosity, we are going to have classification of based on the length. So each length of the runs of homozygosity, we're going to have also a coefficient to see the impact on the phenotype. The traits evaluated by the association were birth weight, Winnie conformation score, Winnie hair coat, post-Winnie weight gain, yearly conformation score, yearly hair coat, scrotal circumference, ribeye area, subcutaneous fat thickness, rump fat thickness, intramuscular fat, and tick count. So all these traits is currently having been considering in the official evaluation by the association, and they give us like this phenotype so we can use for the inbreeding depression. For the inbreeding depression, we use a single trait linear model, regressing the phenotypic values on the estimate of the inbreeding coefficient using the 6 metrics of inbreeding that I talked before. For the genome-wide inbreeding depression, what we did was code the animal genome to indicate the presence or the absence of the runs of homozygosity that we identified before in the study. This followed the work that was proposed by Price in 2014. As we are using some of the traits repeated measures, and we're going to use the GCTA software, we need to regress the EBVs, so to use as the upset phenotypes. And using the GCTA, we also, through the mixed and linear model association, we also use 3 types of different relationships between the animals. The additive one, based on the runs of homozygosity, and also the combination of additive and runs of homozygosity. So the first results that we're going to observe is the average of inbreed for each one of the metrics. We can see here that for the metric that's showing the highest inbreeding, average inbreeding coefficient was based on the runs of homozygosity, and somehow this is expected once the runs of homozygosity, they only not accounting for the current inbreeding happening in the population, but they also are going to account for more historical inbreeding that came in the older generation. Interesting point to also mention here is that for some metrics we observe negative values, and this can be interpreted as a gain of variability for some animals inside of the population when they compare it with the base population. Another point here, we also can classify the inbreeding based on the length when they have inside of the population. For example, as I mentioned before, the segments that have smaller or than 2 megabases, they are normally associated with 25 to 50 generations ago, while in the longer segments, they are going to be more related to from inbreeding happening in the population in the current 3 generations ago or in the current population. The pattern of the inbreeding over time also was different when you consider each one of the traits. And this difference is somehow expected once we are talking that each one of these methods and concepts, they capture different types of inbreeding. What we saw that when we consider just the pedigree is actually that was a decrease in the levels of inbreeding over time and an increase of the number of animals inside of that was registered. That may happen because of we are assuming that along with these animals that are inside of the was registered, they are unrelated, and that is not true. In somehow these individuals they were related. And when you talk about the inbreedings, you can see the inbreedings going up even for the metric, just considering the pedigree, just for considering the genotype animals that were actually increasing. Methods also based on the runs of homozygosity that was increasing over the time. The relationship among animals was decreasing. So these differences that we are capturing during the time, they are reflecting how the pattern over the inbreeding increasing or decreasing over the time. And each one of these metrics, they have their particularities. So one of the metrics that they are more linked to the dependency, linked to the correlation, the gain or loss of the variability, they also are going to be depend of the allele frequency. So these metrics that rely in the allele frequency, they might not function correctly if they differ from the frequency differ from the base population, from the fauna population. Also, some of these metrics, they are going to give more weights for rare alleles, where we're going to so homozygous for rare alleles, animal is going to have a higher coefficient. If they have a rare allele in homozygous, then another individual that doesn't have a rare allele. All these particulars, they are going to affect also the correlation among the metrics. And the difference is also related to these correlations. They also is going to be related to what they are considered as inbreeding. We could observe that even negative correlations was found between, for example, the relationship matrix and segments of the runs of homozygosity, or even just consider the homozygous inside of the population. In the other parts, metrics that are considering in genomic metrics, for example, homozygous, unit gametes, and runs of homozygosity, they had higher correlation among the metrics. With that, what brings to us is, what is the best method? Well, the answer, we don't know yet. What we know is that the best method is going to depend from the intention. So, for example, when we have finite and small effective population size, it seems that runs of homozygous can be one of the best approaches to study the inbreeding inside of the population. But if you want to talk more about losing in or variability, maybe the other metrics can also give some beneficial for the studies. When we talk about the inbreeding depression by itself, what we saw is that the majority of the effects majority of the results were unfavorable for the majority of the metrics, with the rump fat thickness have the highest impact in increasing 1% of the inbreeding. So we could see that we decrease the production in minus 1.13% with the increase of the inbreeding. Interesting, we also observe positive effects in the inbreeding. So in birth weight, when we have considering the smaller lengths, and also in the intramuscular fat when you consider the metric for homozygous concentration. And one point that I think was super cool in this point is that in same trait, we are able to observe positive effects when you talk about a smaller length and negative effect when you talk about the higher length or bigger length of runs of homozygosity. This difference between the across their race class exemplified in one of the relationship between the inbreeding and also trait performance. So maybe this could be more related to the selection of a time that for beneficial, and now you're gonna see something more the fact when you have longer runs of homozygosity. For the genomic, the inbreeding, genomic-wide inbreeding runs of homozygosity, for the majority of the traits, we didn't find any significant region linked to the phenotype. But for tick count, what we observed was a specific region when we use the runs of homozygosity relationship, that region was shown to be significant for the association of the inbreeding with the trait. Looking for the for this region, this region is in the chromosome 8. What we found that is 3 genes that are related with the linked to this region, and they are going to be more related to the apoptotic process. That is the programmed cell death. And have been, like, if you look in the literature and we're talking about resistance for tick, is some process that can be, that can help in the resistance. As conclusion, so we found significant variation in breeding coefficients across different metrics, predominantly favorable effects of inbreeding several traits. The runs of homozygosity associations study identify significant markers linked to homozygous states for tick count, and the choice of the relationship estimate method can substantially affect the interpretation of the genomic association. So I finished to talk about the work that I've done here in this last year. I'm going to talk a little bit about our group here and present what we have been done here at Purdue. So the Rojas Lab started in September 23 under the supervision that Dr. Inaya Rojas. In the beginning, we have just 2 students, master students in the time, Ayu and Adebizin, and Gabriel was the postdoc that was helping him. In 2024, I entered to the lab. And today you have a different composition. We have Ayo and Abiz now they are PhD students. Huanji or Taimin, he is also a PhD student. Yufeng, he is a master student. Rieko is our undergrad. Milen and Rafael, they are visiting scholars here with us. Rafael actually just returned to Brazil. Milena is also from Brazil. She is from the Federal University of Bahia. Rafael is from Federal University of Paraná. And Gabriel, although he's now working in the Interbull, we all always say that once we are inside of the Rojas Lab, this is for forever. We don't leave. Even if you leave, you're always going to be part of our group. So the first project that we're going to talk about a little bit is the project that Gabriel and Adebise was work together with the ABCZ. So they are responsible to perform the genetic characterization of Nelore cattle, where Gabriel he was working more in the genetic characterization imputation process. They were evaluated 300,000 genotype animals. And at the BZ, she was working with genome-wide association study using the APY, algorithm of Provian and Young, in 16 fission traits evaluated by the ABCZ. And right now she is working with the development of personalized SNP panel for ABCZ. Still talking about Nelore, we have Milena. Milena is our visiting scholar, as I mentioned, from the Federal University of Bahia. She's worked with another population of Nelore from GNCs, where her studies more linked to the morphological defects in the Nelore breed, where she evaluates genetic parameters for 11 defecting traits in Nelore cattle, and also doing the comparison between Trashon and linear model to estimate the ABVs, and genome-wide association stands for 11 defect traits. Ayo, he is our PhD student here. He is working with Angus cattle together with the American Angus Association and the Australian Angus, where his studies is linked to the modeling approach of genetic evaluation mature cow weights. One of the approaches is using recursive approach for that, account for body weight. Also perform genetic parameters for mature cow size, and genomic-wide association sets for mature cow weight and body condition score. Taimin, he is work with genomic diversity in genetic parameters in Chinese pigs with a Chinese company that contacted us to help us to develop their breeding program. So the first step that he's doing right now is to see the comparative genomic diversity of Duroc, Landrace, and large white pigs. And also later on, he's going to perform genetic parameters of different traits evaluated by the company. More in the molecular side, we're going to have Rafael, that he was working here at us with the use of the convolutional analysis in sequence cell RNA-seq. So he was testing different methods, softwares, and reference for the convolutional analysis. What we saw is that the software and the reference impacting the results. And now he's working in the convolutional analysis in the book RNA-seq data of longissimus dorsal muscles biopsy of Holstein cows. We just wrote and gonna talk more about this analysis on the SCA. That is gonna be one of the oral presentations. And one thing that we have been talking a lot and have been proud about the results that I have done here is about sustainability in livestock production. On specific in this topic, we have Yufeng work in his master, and Rieko also that was finishing his as an undergrad and probably come in fall as a master student. So our idea here is enhance methane phenotype together with the people from the engineering department, developing a novel in vivo robotic rumen bolus sensor for enhanced phenotype in methane emissions from cattle. We already have preliminary results based on the in vivo sensor and the sniffers, but the main goal someday is to develop this robotic sensor that can be swallowed and stay in the rumen of the individual for the entire life, measuring methane in real data and in real time. So here, just all our companies and associations that we have some type of partnership, and we're gonna say thank you for letting us work with you guys. And also, I would like to say thank you for each one of our members and our groups that have been working in so different projects and have been successful in these projects. A special thanks to Dr. Rojas that have been conducting and leading our group. And thank you guys for listening, and I'm open for questions.
Thank you, Henrique. We'll now open the floor for questions, either from the room or online. Yeah, go ahead.
So, Henrique, first of all, congratulations for the work you have done and for the presentation. I would like to ask you something about your principal components analysis you presented. The American Red Angus was a group apart of the other cluster. And in the other cluster, you had Australian het and black Angus, Brazilian het and black. So the red Angus from Brazil and from Australia, and I guess also from Canada, I'm not sure, they grouped with the black Angus and not with the red Angus. So,
exactly.
I was thinking, could be due to in these countries they allow some type of some level of crossing between red and black Angus, and because of this they group with the black group and not with the red.
Yeah, good point. We also were thinking about that because when we mentioned the first time that we have the black and Angus together, we are expecting that the red is going to be with the red Angus association, the black with the black. But what we also need to account is that the red Angus over the time, they start to have their own decisions on the selection index. And so they start to have their own process of selection. So I believe in more point of the view that as the selections for this population was mainly specific for the side of this population, this could drive differentiation what we observe for the others. Because for the Brazilian in the Canada, I don't think we have a split in red and Angus about the selection process. They are just the color of the animals. I don't know if I'm not, this is just my, I cannot like prove, but say with sure, but I believe so, that's like that. So all this differentiation that we see is more related to the selection process that happened inside of the population over the time, being more closer to this population. Did I answer your question?
Yeah, thank you.
You want to go first and then we'll go.
Yeah, mine is related, but go ahead. Yeah, I was just following up on this question. So if you have red Angus in the United States as separate, and let's say you're a red Angus breeder in one of, let's say Canada or Australia, and you use animals from that red Angus population in the US, how do you handle that in a genomic evaluation if there's lower correlation in gametic phase? Like, how do you make sure that the evaluations are correct? Can it be just due to higher density, in your SNP genotypes? Does that handle it, or is there any special things we need to do to make sure that we get the phase right?
Yeah. So to make sure, like, as the correlation, the correlations, yeah, I see. To make sure that if you're gonna put the red, I'm gonna say that we need to have like more dense panels or more panels markers in common that is gonna be in high linkage equilibrium between the KTL, and they are in common inside of this population. So we will be able to have a higher correlation in the geometric phase for that. Based on the panels that we have for each one for the population, if you merge everything, and the results that we're showing here is that these could not give us so accurate we could not predict so accurate the breeding values of the animals using the information that we have right now. So a more higher density is going to be, or increasing the numbers in markers in common than they are linked to the KTLs could be one of the options.
And I now have two questions. Thanks, Henrique, for the presentation. Very nice. My first question for you is regarding to this study. Why you didn't do admixture analysis?
Interesting point. Like we were like when we discussed it was something that I proposed and we're like, I don't know if you're gonna find anything because all they trace back in the same origin. So it's something that we could include, yeah. So we're gonna see the influence of one population in another, especially for Brazil. But also like
it's not only between them, but with the cows that they use as a base. When they imported the sires, they usually didn't have an Angus cow. They use other breeds. So, yeah. And with that mixture, if you have, if you go small enough, so the density of the panel is small enough in the chromosome, the genome, the regions, you can see that mixture. True. Even though you don't know which breed it was, but you are going to see a genetically different group. So then that will be interesting to show that, let's say, in Canada, the mixture with the existing breed was different compared with the United States or Australia, because it's not going to capture just between the Angus, but what was the Angus, the side Angus used on when they imported.
Yeah. Yeah,
and that could explain some difference that you are seeing
on the results. True. Yeah, it's something that we can perform. We have the material. And it's something that we can consider and perform. Thank you for the suggestion. We are not thinking about that, more about the influence in each one of the population. But yeah, this maybe could help to explain a little bit more about the history.
Yeah, I forgot now. There was another question, but I forgot what. Oh, okay. Now I'm going to change the subject. So let me ask you something, Henrique. If you have a change in homozygous over time in a population, why the change could happen? What's the phenomenon that would lead to increase the homozygous?
The runs of homozygous.
No. Not the runs. You can rule, but the homozygous of the population.
Yeah,
they can happen. With the time they get more homozygous.
The most common reason is during to the selection about like when we talking about inbreeding, but is one of the factors.
What was the fact?
The inbreeding, when you're talking about one of the factors, you could talk about selection. Yes, naturally.
Yeah, selection, but could be drift, chance.
Yes, drift, yes.
Right. Mm-hmm. But all this selection and drift, they cause inbreeding. And that's the reason that you increase homozygosity. So if the increase of homozygosity is due to inbreeding, so the only way to the homozygosity to increase is because we are making relatives. Mm-hmm. right? So the increase is due to identical by descent,
Mhm.
not by identical by chance or state.
Yep.
So now my question to you, what definition of inbreeding do you think is more important? The right or the molecular definition?
Good question. More important. So the molecule is going to be more linked to that because it's direct to the homozygotes that came from the inbreeding by descendant.
Exactly.
Simple.
And that homozygous by identical by descent that changed over time. And the mechanism is we are making relatives, or because we are selecting, or because it's a small population by chance, by drift, you are making relatives. There is no other mechanism. So molecular is the definition to think about. So when you measure runs of homozygous, you are not measuring homozygous that is due to only to the identical by descent. You are capturing the entire homozygous. That could be just by being the same allele by state, or because that segment really came from a common ancestor. So that's why you have a higher inbreeding, as you mentioned, right? But the change over time of the runs of homozygosity, that change should be due to identical by descent. And the same for the other statistics that use both identical by state and descent, like the whole homozygosity. What's important is the change over time, not in a specific year, right? It's the change that tells you this homozygosity that's caused by inbreeding, a change, right? So I think that's an important thing for us to keep in mind. And that's why most of when you talk about inbreeding, we talk about molecular, that definition.
Yeah, and one interesting point that even when you talk about runs of homozygosity is that the metrics now, we're still discussing about the parameters how to use, exactly because of that. Because theoretically, runs of homozygosity should be just, runs of homozygosity should be just identical by descendant. But you're not able sometimes to identify just that. There is some softwares that they do the phasing first to account for that. But what they mention is that sometimes we need to be more strict in the what you call runs, decreasing or putting a limit of what the minimal size of the run. When you put, for example, in bovine, we are talking about, I think, 500 kbs. That is going to eliminate a lot of the identical by chance and just have a more idea what it could be the identical by descendant. But yeah, I totally agree with you. Is a really good point.
I think that the majority of papers and studies, they don't separate. It's just common homozygotes in that region, right?
Yeah.
Is there another question? Is this time
At the end? Okay. We'll go to Alan online if you want to ask your question.
So the question one would be, how serious is the potential inbreeding in the red Angus? Are you very concerned?
The red Angus?
Yes.
They didn't show any type like differences when you talk about compare with the others. So not that it could show us something to pay more attention.
Have do they have more runs of homozygosity and big runs of homozygosity?
They have more amount, but it also this is also one thing related to that their density of the panel was a little bit higher. So and it was more linked to the small length, not so much the longer length. I think Brazilian was the one that has the largest runs of homozygosity that we found in the study.
Because red Angus just don't have as many AI sires to sample or as many total animals. But you could, I think that the red Angus still have an open herd book.
Mm-hmm.
and you can still bring in black Angus, but you'd have to back cross them twice, and then half the animals would be red again. And you could always add a little bit back from the black Angus, but you'd have to give up red matings to do that.
Okay. Hey, I'm gonna check that, Dr. Shinko. It's a really good point.
All right. Do we have any more questions in the room? Shelby?
I was just wondering if you have any idea or a theory as to why recent inbreeding is higher in American and Australian cattle versus like the other countries.
Can you repeat again? I didn't understand the.
Yes, no worries. I'm just wondering if you have like a theory or like an idea as to why more recent inbreeding is higher in Australian and American cattle as opposed to the other countries.
Oh, for America and Australia?
Yeah.
Comparing to which metric? Sorry. For the runs of homozygosity, you mean?
Yeah.
That is if you see, I think it's gonna be more related to we have more intensity happening in these two populations, and they are more linked. So it would be nice to see like how even for Canada, so we can see the three of them together, and if they are comparable when you talk about that. But like Brazil is young yet, is about the association and how they work as evaluation. So I think it's going to be more related to that, to the selection press over the time.
Thank you.
You want to ask your question?
Yeah.
Okay.
Yeah, no, Henrique, I really like when results that you show there that you're showing that not all inbreeding is bad, you know. And especially when you look at old inbreeding, those small segments of homozygosity, the genome, you show that they have a positive association with a trait.
Yeah.
And then when you look at longer segments that are more recent breeding, you show depression, negative results. But that's interesting, right? Because we expect that. Because the old inbreeding, if it's still there, the region, because you fixed something that was favorable.
Yeah.
Right? And therefore it would be positive for the trait. But the recent inbreeding, you could have a mix of favorable and unfavorable, and even more unfavorable, because there was no time for the selection to purge. I think that's what the word for I got, purge. That is deleterious of alleles and alleles that are not favorable. So I think you have to keep this in mind, right? That not all inbreeding is bad. And if you fix regions that are favorable for what you're aiming for, that's good inbreeding. But of course, it's the side effect of inbreeding, recent inbreeding, that we expect depression. But with time, if you have a strong selection process, we are going to fix again alleles or combination, epistatic effects. Don't forget about combination that are favorable for the particular breed. So I really like to see this, David. We did the similar study in our group by Odi, that we showed exactly the same with the Derek, looking at this old and what we call old and recent breeds.
Yeah.
Very nice. Yeah.
Can I make a comment about this? And a question for the whole floor. Wouldn't that specific inbreeding so was favorable, the oldest one with birth weight, but now the newest one are unfavorable? Couldn't be do a different direction in the selection? So we are trying that to select that population for smaller animals to make the parturition easier, because the animals were getting higher, and then we shift the direction to get smaller animals when they give birth.
Well, I think for the analysis, it should not be have this confounding. effect. I think it's not the reason. The reason is because it's more recent breed. It was not time to counter any negative effect because the selection didn't have time yet.
There was probably unfavorable older inbreeding. It just got eliminated over time.
Oh, yeah.
Okay. Okay.
Yeah. There was time to eliminate.
Mhm.
And then we just kept the good, what we call good inbreeding,
Okay.
the regions that are favored.
You know, there's an argument to be made to do more of these kind of studies because we have a lot of breeders that get worked up about inbreeding, but it's just a number. Inbreeding depression is what we really should be worried about.
There for today. Thank you so much, Henrique, for your presentation. It was a great presentation, great discussion, and some interesting questions. We will be posting this presentation to YouTube, and we'll share all that information with you as well. And we'll see everyone back here again next week for another presentation.
Okay. Thank you for
Yeah.
having me.
And this, Mike, this discussion is even more relevant nowadays because of